Hi, I'm Sarah Camp. I'm going to be talking about my senior research project on the inhibition of rhizobium growth by organic certified pesticides. I'm going to start by reviewing the transition from synthetic to organic pesticides, talk about my methods for my experiment, go over the results that I received, and present future research studies based on these results. Conventional pesticides are used to increase agricultural efficiency by preventing plant loss due to insects, fungi, and competing plant species. Pesticides may present a threat to human and ecosystem health, in part due to the overuse of pesticides by farmers over the recommended agricultural dosage and the accumulation of these chemicals in the soil. The pesticides can run off into streams and water sources and negatively affect non-target species through direct contact. The threats of synthetic pesticides have led to a movement towards organic certified pesticides. Organic agriculture, according to the USDA, is an ecological production system that promotes and enhances biodiversity, biological cycles, and soil biological activity. The field of organic agriculture is increasing greatly. From 1999 to 2000, as seen in this graph, the land usage for organic agriculture worldwide has increased fourfold in millions of hectares. There's little research done currently on the ecological impacts of organic agriculture and of the use of organic certified pesticides. Because of the little research, there's also little restriction, and many of these pesticides need to be applied more often than synthetic, which can lead to a greater accumulation in the soil. What research has been done on organic certified pesticides has shown that there is some negative effects on non-target species. Mineral oil and Botanigard, for example, have been shown to be less effective and less environmentally sustainable than their synthetic counterparts. Toxicity studies have also been done and shown that some of the most toxic pesticides are organic. One of the main areas of concern for the ecological effects of organic pesticides is the rhizosphere. The rhizosphere is the ecosystem in direct contact with the roots. Shown in this picture, you see a root system and a lot of different colored dots. This represents the microorganisms that drive the rhizosphere ecosystem. These microorganisms work directly with the plant to help with nutrient breakdown as well as nutrient uptake, and are extremely important for plant and crop health. Rhizobium is one of these species. It's a plant growth promoting bacterium that fixes nitrogen in the plant as well as inhibits the colonization of roots by potentially pathogenic species. In the past, conventional synthetic pesticides have been shown to inhibit rhizobium growth. I aim to increase the data about organic agriculture and to determine the impact of organic pesticides on rhizosphere health. Because organic pesticides have been found to negatively affect non-target species in the past, and because pesticides have been shown to inhibit rhizobium growth, I hypothesize that organic certified pesticides likewise inhibit rhizobium growth. For my study, I used Rhizobium leguminosarum, which is a fast-growing rhizobium species, which is also plant growth promoting and nitrogen fixing. It is found in root nodules and free in the soil. I subjected this culture to 4 pesticides. I used an organic insecticide containing spinosad, which has an agricultural recommended dosage of 0.01%, and compared this to a synthetic insecticide with permethrin, which has a 2% recommended agricultural dosage. I had also looked at fungicides. I looked at a organic containing copper octanoate, which is a copper salt soap fungicide, with an agricultural dosage of 0.08%, and compared that with a mycobutanil fungicide, which is synthetic and has an agricultural recommended dosage of 0.02%. For my experiment, I conducted an inhibition zone assay in which I inoculated plates with 10 to the negative 2 dilutions of Rhizobium leguminosarum for 24 hours. I placed 4 paper discs on each plate, 3 with 1, 2, and 4 times the recommended agricultural dosage of the respective pesticide, and a 4th disc with no pesticide and only water, and I used that as a control. I grew the plates for 48 hours at room temperature, and I measured the diameter of inhibition around each disc after these 48 hours. I also did a growth curve assay in which I created optical density growth curves using an electronic plate reader of 10 to the negative 2 dilutions of 24-hour Rhizobium culture. I subjected this culture to 1, 2, and 4 times the recommended agricultural dosage of each pesticide, and I included a sterile water control, a broth control, as well as a blank to correct for absorbance with only broth and with broth and pesticide. I read the plate at 630 nanometers for 24 hours, and I used the absorbance reading to create the curves. For my insecticide inhibition zone assay, I found that my organic insecticide with spinosad did not inhibit Rhizobium growth. That is the picture on the top here. You can see there is no inhibition zone around the 3 discs with pesticide, and also no inhibition zone around the control disc. When compared to the synthetic insecticide with permethrin, however, there is a large difference. Permethrin did inhibit Rhizobium growth. In the picture here, you can see the darker regions around the discs with pesticide. The graph here shows the dose-wise response in inhibition to increasing concentrations of permethrin compared to the very minimal inhibition by the spinosad. The fungicide inhibition zone assay gave completely different results. My copper octanoate fungicide, which is organic, was found to inhibit Rhizobium leguminosarum growth. It actually has greater inhibition zones than the synthetic mycobutanil pesticide that it was compared to. Although there is no statistical significant difference between the organic and synthetic pesticide, there is a significantly higher inhibition from the control, which was at 0 inhibition. You can see again the dark zones around the 3 discs with pesticide in both the copper and mycobutanil pictures here. And there is also a dose-wise response with increasing inhibition to increasing concentrations of both pesticides. For my growth assay, which I haven't been able to collect the data yet, I expect for the copper fungicide to have a greater or equal lag to its synthetic alternative, which will be looking at the smaller or equal slope on the log phase of the curve. For the spinosad insecticide, I expect it to have less lag and greater growth than the permethrin synthetic alternative, I expect it to have a greater slope than the synthetic alternative. I will plot my absorbance data on a log scale versus time, and I will use the slope of the log phase of the bacterial curves for my comparisons. Based on my inhibition assay, I can conclude that some organic pesticides do inhibit rhizobium growth. This is most likely dependent on the mode of action. The copper octanoate works by denaturing cell proteins, which would affect bacterial species and did in my experiment. And the spinosad works through an excitation of the neural system, which causes paralysis in insects, but would not be expected to affect bacterial species. The dose-wise increase in inhibition with concentration in each of the pesticides shows that an accumulation of these pesticides would cause an increase in inhibition. So accumulation in the soil in field would cause an increased inhibition of rhizobium growth and decreased health in the rhizosphere. This has implications to farmers who need to realize that they need to stick with the recommended agricultural dosage of whatever pesticides they use, and keep in mind that just because a pesticide is certified organic does not mean it's necessarily 100% safe for the ecosystem. Policymakers should reevaluate organic agriculture and treat organic pesticides on a case-by-case basis in making regulations as they have done with synthetic pesticides. And consumers need to realize that just because something has a label organic does not mean that it's necessarily environmentally sustainable, like the organic definition says. In future studies, I would like to see on-site analysis of the actual concentrations of organic pesticides that have accumulated in the soil. I would like to determine the ability of the soil to filter these chemicals, and I would like to see a study like mine conducted in the field over the inhibition of rhizosphere microbes by organic pesticides in its usual environment. Thank you. This is the end of my presentation. Have a great day.